So welcome everyone for this Neurobinar of February. I am Ingrid Meucci, the Communication Manager of Neuro Marseille, and I'm pleased to welcome you here. Just a quick reminder, we have a 1 hour conference and we will begin with the cross presentation of 2 PhD students. Each will present the PhD of the other. Then there will be 5 minutes of questions, followed by a presentation by Pierre-Yves Jacob for 20 minutes, and then 50 minutes of questions. Exceptionally today, after 5 p.m., we will welcome Justine Faccini to present the Student Society AG3C for PhD students and postdoc. So do not hesitate to stay connected to find out more. So today we are pleased to welcome Lucas Goirand-Lopez from Inmed. We will present Dominic Bingham's PhD on the role of actin at the presynapse, co-directed by Christophe Leterrier and Marie-Jeanne Papandreou, sorry. Then Dominic Bingham from INP will present Lucas' PhD on impact of kainate receptor on the network activity of the epileptic dentate gyrus in silicon study by modeling, co-directed by Valérie Kreppel and Thomas Marissal. Finally, Pierre-Yves Jacob from LNC will present his team and its research on the brain network underlying spatial navigation in complex environments. He will be introduced by Boris Bull. So Lucas, it's up to you to start.
So hello everyone, and welcome. I'm Lucas and I'm going to introduce you the thesis of Dominic Bingham from Neurocytolab from INP at Latimon. And Dominic is working on the organization and the role of the actin at the presynapse. So why is Dominic interested in the actin? The actin is a common protein involved in several processes in the body, but its function at the presynapse is still in debate and controversial. So Dominic starts with the hypothesis that different presynaptic actin structures will have distinct roles, and his aim is to define their structures and to better know their function for the presynaptic organization and the presynaptic physiology. So how is doing that? To look at the actin is using a lot of imaging, but to be sure to not look at postsynaptic actin is using a solution based on polylysin coded beads, and that beads will induce a presynaptic differentiation when contacting an axon. So here you have an example of the beads with all of the presynaptic components. So they will see that thanks to fluorescence. And Dominic and its team validate the model and show that 80% of the beads that contact an axon did induce the formation of a presynapse. So only 20% aren't creating any synapse. And among them, 70% are enriched in actin. So we will call this category A+ for actin enriched, and the other A- for actin non-enriched. So as I said before, they look at the presynaptic component, but they need also to have a functionality measure. So they are looking at the vesicular recycling that is linked to the presynaptic activity. And Dominic is using the synaptotag-mean antibody to have fluorescence to measure this vesicular recycling. So what did they see? They see that the actin-enriched one have more presynaptic component and show more synaptic recycling than the beads with the non-enriched beads and the beads that aren't creating any synapses. Here you have 2 different bars because they are just using 2 different ways to induce presynapse activity. And after that, they look at global perturbation of the actin, but both increasing and decreasing perturbation will lead to a reduction of the synaptic recycling. That suggests that you really have an importance of the structure of the actin in the synapse, in the presynapse. So to better know about it, Dominic is did some subtle perturbation. So he's going to perturb two different structure of actin. So the branched one is inhibiting the branched one is showing a little increase in presynaptic components and the light increase in synaptic recycling. Those increase could suggest that the branched structure could have an inhibition effect on the vesicular recycling. Whereas when he inhibited the linear actin, he showed that he have a significant decrease in presynaptic components, but also a significant decrease in the synaptic recycling. So that could suggest that the linear structure is helping that synaptic recycling, the vesicular recycling. Sorry. So to conclude and resume, Dominic and his team did some pretty cool and useful jobs, and they show that the presynapse can be separated into two categories: one with actin enriched, and the other not the actin non-enriched, and that the actin enriched are linked to a better vesicular recycling. And as the global actin perturbation always lower the vesicular recycling, Dominic and its team look at more specific structure effects, and they could suggest that the branched actin structure could have an inhibiting effect on vesicular recycling, whereas the linear one is helping it. To further investigate it, Dominic wants to use a more specific model of perturbation on actin structures, and he will follow that thanks to super resolution imaging that gives him some super cool image, but also super useful because he's going to see that structures in reality. So I'm going to finish with some photos of his teammates and collaborators, and thank you for the listening.
Thank you, Lucas. So Dominic, you can go, and do not hesitate to already ask your question in the Q&A section. Thank you.
I'll just share my screen now. Can everybody see okay?
Yep.
Yep. Okay. Well, just before I begin, I want to say thank you to Lucas because he did a really good job there presenting my work. Yeah, this is the Zoom thing. There we go. So yeah, I'll begin. I'm Dominic Bingham, and I'm presenting the work today of Lucas Goirand-Lopez. And as you can see, his project is titled Effect of Kinase Receptors on the Activity of the Epileptic Dentate Gyrus Network, and it's an in silico study. Lucas is in the neuronal coding and plasticity team, plasticity and epilepsy team, and he's supervised by Valerie Kerpel. So there we go. So what is Lucas investigating? He's looking at these ectopic kinase receptors and their impact on temporal lobe epilepsy. So just to kind of give some background, temporal lobe epilepsy is the most prevalent form of epilepsy, and it represents approximately 70% of cases, and around 30% of these are actually resistant to therapies. So really a better understanding of TLE is fundamental in order to find new therapeutic targets and ways to treat this disease. And the area that Lucas is looking at is the dentate gyrus, and this is located in the hippocampus. And one of the main features of the epileptic dentate gyrus network are these recurrent excitatory circuits. And to a layman like me, they're just kind of excitatory loops. And basically, these lead to aberrant and random massive synchronous network activity. The synapses of the aberrant connections present a particular kind of receptor, i.e., unsurprisingly, these are the kainate receptors which Lucas is looking at, and these are absent actually in the healthy dentate gyrus. Now, of course, a better understanding of these of the effects of these receptors may help us to use them to find a therapeutic target for TLE. And to investigate it, Lucas is really going to compute an in silico model of the epileptic dentate gyrus. And within that, he hopes to contain these kainate receptors. So the computational model. So the computational model is split into two components, the first being the cellular model and the second being the network one. And each different cell in the cellular model is biophysically based with a membrane activity, and that contains capacitance and ionic exchanges and potential conduction as well. And each cell will be defined by its physiological properties, channel densities, and shape parameters. To create the network, cells are going to be connected according to biological observations here. And he also represents synaptic connections in this model. Now, of course, the network model used is going to be reduced to scale. And as you can see in the top right, it's reduced to approximately 1 in 2,000. And within this network model, Lucas is going to use the 4 most prevalent cells in the dentate gyrus, namely these excitatory granule cells, inhibitory basket cells, excitatory mossy cells, and also inhibitory HeLa interneurons. So basically, the difference between the control and the epileptic model, and it's good there is a difference between these two, is that the network is changing in epileptic conditions in terms of the presence of aberrant connections between these granule cells. And these are really well illustrated in this image in the bottom left. So as you can see, these connections between the granule cells are labeled MFS, that stands for mossy fiber sprouting, and these arise from cell loss in the helix. So Lucas's PhD aims to add a model of these ectopic kainate receptors at the synaptic connections of these mossy fiber sproutings. So the computational model with these kainate receptors. As you can imagine, the modelized kainate receptors have to have the same properties as the biological ones. And biologically, kainate receptors represent half of the glutamatergic receptors at mossy fiber synaptic connections. Also, the evoked postsynaptic potential, or EPSP, from biological kainate receptors have slower kinetics than those of the AMPA receptors. So to briefly explain, an EPSP is an experiment where you can stimulate the synaptic terminals with a pulse and look at the postsynaptic potential. And then basically, when AMPA receptors are stimulated during EPSPs, they show no significant difference if the membrane is at resting potential, so minus 70 millivolts, or if it's depolarized at minus 50 millivolts. Lucas's team also looked at the spikes generated by these EPSPs, and these spikes are basically represent like single neuron events. So the epileptic model presents these longer lasting plateaus, and you can see this on the diagram on the left. And these are because of the kainate receptors. And then these plateaus, these lead to jittered spikes and loss of functional synchronicity. And this is shown on the right in really kind of beautifully in the sort of Joy Division album cover looking diagram on the left there. So Lucas, of course, is still completing his PhD. So as you can imagine, the kainate receptor modeling is still in progress. And when the properties of this modelized receptor will be satisfying enough, network activity will be launched by the preferent path, which is the kind of path into the hippocampus and dentate gyrus, and then be analyzed. The in silico experiments will let Lucas see the effects of the kainate receptors on network activity, and these will be under different parameter options. And then he'll be able to create experiments with perturbations or changes in the kainate receptors, and then he will be able to work to see if he can use these receptors as a therapeutic target. Now, also, he'll be able to compare these results with in vitro experiments to better investigate some like maybe kainate receptor blockers and their effects on biological tissue. Now, of course, Lucas would like to thank his team because they've done some really cool work here. And I would like to thank you all for listening.
Thanks everyone, and thanks Guy. So you can begin to ask your question in the Q&A section, or you can raise your hand to ask directly your question. And Dominic, you are the first one to begin. You have one question.
Where is that question?
In the Q&A, yeah.
Okay. How come the difference in actin structures influences so much presynaptic activity? So if I think I've understood the question, basically we think that actin is so you've asked for the difference in the structures. We think that actin is actually representing a lot of different structures in the presynapse. I actually showed the image at the end, shown kind of some of the structures will be like we think might be there, and I kind of hinted at it with the mesh and rail structures. So we think a lot of just the kind of machinery for exocytosis is kind of at least structured with actin. So maybe there is going to be the mesh-like structures which can inhibit vesicular release, and therefore, you know, if we inhibit them, we might expect a slight increase in vesicular recycling. Or maybe with those rail structures that I talk about, these help bring vesicles to the cell membrane to be released. And therefore, obviously, when I inhibited them, that might explain why there was a decrease in vesicular recycling. And so how come the difference? Yeah, I think I answered the question.
You have another one, Dominic?
Okay, that's an interesting question. Because, well, one, I've pretty much used the roughly the same size beads.
Sorry, Dominic, could you just read the question for everyone? Because I don't know if everyone can see it.
Okay.
Thank you.
Yes, it says, is the presynapse formation, brackets, number or size, and function depending on the size of the beads used in your experiments? So is the presynapse formation and the function depending on the size? Yeah, so we've actually used pretty much the same size beads for all of our experiments, but that doesn't mean we get the same size synapses. And also, it doesn't mean that, like it has to be said that when we have these non-enriched and actin-enriched, these aren't just large synapses that have a lot of actin, small synapses that don't have a lot of actin. We see a lot of variation in the different presynapses that we look at. So I'm not sure what the effects if we use the larger bead, but the reason we use the beads we do is because they're kind of just appropriately sized to kind of fit nicely to the side of an axon. Like if they were too small, maybe they wouldn't induce a presynapse, and if they were too big, then they might just crush the cells or something. I don't know.
Thank you. Well, maybe waiting for one or two questions more for Lucas. Okay, I think your presentation was so clear. There is no question. So thanks everyone. And we have one hand.
I did not understand much the purpose of that receptor. If he could explain it better, that would be nice. That's not a problem.
The kinase receptors?
Yes.
Yeah. So it's just my team and other person from the labs show that in epileptic condition, at the MOSI fiber sprouting, so in aberrant connection, we don't just have the AMPA receptor, that is normally in a healthy condition, you have AMPA receptor in the dentate gyrus, and we have half of the glutamatergic connection, aberrant connection, are present these kinase receptors that isn't, and you don't have any of them in healthy condition in the dentate gyrus. So that's why we want to know what the effect of that specific receptors in this network. I don't know if I answered well your question.
Thank you very much.
Thanks, Aya, for this question. So do not hesitate if you remind something about the presentation of our PhDs to ask your questions. They can answer them later. So we're going to go on with Pierre-Yves Jacob, presented by Boris Burle.
Hello, everybody. It's my pleasure today to introduce Pierre-Yves Jacob, the speaker of today's seminar. Pierre-Yves' initial training was in sports science, and after a couple of years as sport teacher in secondary school, he came back to the university to follow a bachelor in neuroscience and physiology. After a master in neuroscience, he started a PhD at the LNC under the supervision of Francesca Sargolini that he defended in 2014, working on the neural basis of spatial cognition. He then moved to the UCL in London in the group of Kate Jeffrey for a postdoc from 2014 to 2017. After coming back to France, he has been awarded a return grant from the FRM for 3 years. This grant has, however, been interrupted by his recruitment as a CNRS researcher in 2019. During his thesis, he published the first French paper, and so far still the only one, on the famous grid cell. And during his postdoc at UCL, he discovered a new type of neurons, the flip cell, that he will probably talk about today. Pierre-Yves, the floor is yours.
Okay, thank you, Boris, for this nice introduction. I share my screen. Okay, can you see my screen? Is that okay for everybody? Okay.
Yes.
So thank you, Boris, for this nice introduction. It's a great pleasure to be here today, and thanks for all of you to be connected. And I will speak today about my research, which is about the spatial representation in complex environment. So I'm doing my research in the Laboratory of Cognitive Neuroscience, which is headed by Thierry Hasbrouck, Boris, and Béatrice Alescu-Lotier. And this laboratory has now a 12 research team. And I'm working in the team headed by Francesca Sargolini, called Neural Basis of Spatial Cognition. And this 12 research team are supported by 6 technical teams and 3 experimental platforms now. So the common topic between all the research in the LNC is about cognitive neuroscience, which can be defined for me as the brain processes to acquire knowledge and understand our world. So in the team, I work and collaborate with researchers that are lecturer or reader, also professor from the ex-Marseille University, but also researcher from the CNRS. And of course, we have very nice, amazing PhD students and looking for postdoc. So it's a brief introduction about the Laboratory of Cognitive Neuroscience and our team. And the question of the team we try to answer is, how do we find our way in space? And to answer this question, we know the different steps to find our way in space, which is to perceive and to process the information about our environment. And this allows us to create a spatial representation. And I'm working specifically in spatial representation, which allow to locate our own position in space but also to locate the position of the goal we want to reach in space. And once we have created the spatial representation, we can plan and perform navigation. So to investigate the spatial representation, why is it important to investigate spatial representation? Because it's critical for our everyday life as human, or also for many mammals. It's necessary for survival and also to organize our everyday trips and to reach goals. So I try to put some example of condition of necessary for spatial representation, which allow, of course, to go home, which is a condition of security, safety for us and for also rodents, to find food, which are condition of survival. For rodents, it allows them to avoid predatory area, so it's condition of survival. And for a human, it's also a condition of to live in our human society, which is going to work, going to the lab, and to look for friends. So I'm investigating spatial representation by using a technique which is called in vivo electrophysology. So it's a technique in which I record the neural activity when an animal is behaving during my experimental protocol. So I use a frame of electrodes that I implant chronically in the brain. Can you see my cursor? Yes?
Yes.
Yes.
Yeah. So this is a picture I take of the frame with 32 electrodes that I implant in the brain of the animal. And this electrode has in this frame, which is fixed to the skull of the animal. And my animal, my rodent, can live with for months with this frame on his head. And when I want to do my experiment, I place my animal in the experimental arena, and I record the neural signal of my animal with a wire that convey this neural signal to a recording system. And also I track the behavior of the animal with a camera. And when my experiment is finished, of course, I analyze the neuronal activity and also the animal behavior, and I can look at the correlation between the activity of neuron and what my animal is doing. So by using this type of technique, people, researchers around the world find some neurons that are specifically involved in representation space. So I call them space coding neurons. And this neuron has been found in the rodent brain, but they are also in some other mammal brains. So the three main types of these neurons are called place cells, education cell, and grid cells. And in the team, we are working on these three main types of cells. And two of them, place cell and grid cell, has been awarded in 2014 to John O'Keefe, Edward Moser, and May-Britt Moser, the Nobel Prize of Physiology and Medicine. So I will explain all of this cell by explaining, showing some example of this cell, and also presenting some of the preliminary results I have during my project with all this type of cell. So the place cell were the first to be discovered by John O'Keefe in 1970, and John O'Keefe put an electrode in the hippocampus of a rat brain. So the hippocampus is this green structure, and he records the activity of a single neuron, while an animal were moving in this circular open field. And the circular open field is polarized by some object for the animal to be able to move in this space. So I represent the path of the animal with this black line. And as you can see, the animal go everywhere in this environment. And on this path, I superpose the activity of a single neuron. So the action potential are shown in the red dots. And you can see that the red dots are active when the animal is specifically located in this specific part of the environment. That's why John O'Keefe called this neuron place cell, because it's a cell that codes for a specific place. And another way to represent a place cell is with this color code. So I show you because I will use for my example. So the color code is showing the activity of the cell from blue when the cell is not active, there is no action potential, to warm color or red where there is a maximum firing rate. And you can see that the cell has a spatial firing field in this position. So this is a video to show you and to hear a place cell. So I hope it will work and you will not have a lag. So the animal is moving and you will hear when the animal is in this area drawn by the white circle line that the cell is active. Can you hear the sound?
Yes.
Yes.
So the action potential is the tick, 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 you will hear. And when the animal is outside this area, the cell starts to fire. So I use personal data from Vincent Hock to show you the activity that he records of 5 place cells. And you can see that each of them has their own spatial activity, which is different from the other. So the 5 place cells are active in different places in the environment. And if we sum the activity of a small population of place cells, we can cover the whole environment that I tried to show you here with this place field. And this creates this mental representation of space. This is how we can mentally represent our environment. And in the lab, I collaborate with a team that work on food intake to ask the question, does parental obesity affect offspring place cell activity? So we use 2 groups of animals. So we have a group of normal parents and a group of parents that were fed with high-fat diet to induce obesity. And both parents were reproducing to have offspring. And we know that offspring for normal parents have a good memory or normal memory, while the offspring from obese parents has spatial memory deficit. And we wanted to link the spatial memory deficit with the place cell activity. So Manon, the PhD student, record place cell activity from the 2 groups, and she compares the activity, the spatial activity of this cell. And as you can see, for the offspring of obese parents, you can see a nice spatial activity, but the spike density decrease. There is less red dot, less action potential for a place cell from offspring of obese parents. So we believe that this decreased place cell frequency may underlie the behavioral spatial deficit that has been observed before. So the second type of space coding neuron is called A-direction cell. So the A-direction cells are found in many brain structures in the rodent, but the 4 more interesting structures that researchers are looking for are these 4 structures: the postsubiculum and the anterodorsal nucleus of the thalamus, and 2 cortical structures, the retrospinal and the medial entorhinal cortex. And an A-direction cell is a cell that doesn't fire when the animal is in a specific place, but that fire when the animal head faces a specific direction. So we represent a direction cell like this polar plot that shows the distribution of action potential from the head direction of the animal from 0 to 360 degrees. And when the cell is not fired, it's at the center of the cross, so no 0 hertz. And when the cell is firing, we are far to the center of the cross. And you can see that the distribution is maximal when the animal faces in the left direction. And this is just to show you what is a not directional cell that fire everywhere in the environment. So this is another video of a direction cell that you can hear. And you can see that the animal, when the animal faces this white landmark, the cell is specifically active whenever the animal is in the environment. Okay, so in the lab, we record edirection cell, and we can observe that several edirection cells had their activity, which is maximal in different directions. So a small population of edirection cells create a mental compass. And with the activity of place cell and edirection cell, we have all information to navigate in our environment, because the place cell tell where you are, and the edirection cells tell you where you are going, which direction you are going, or which direction you are facing. And with this edirection cell, I investigate the spatial representation in complex environment. Why this topic? Because the example I show you and the property of this place cell and edirection cell were found when the animal move in a single homogeneous open field. And I realized that nothing in our world and in the rodent world is as simple as this homogeneous single arena. For example, in the simple arena, we can have different places that can be associated with different value. For example, this rat can have a place to rest with this nest, or some other place to have food or drink. And also he can create his home base, which is condition of security. And there were some places that are relevant for almost nothing. So one of my questions is, do the space coding neurons represent the relevant places in this type of single open field? And my second question is about connected environment, because our environment were connected usually with door or corridor. So I'm recording this spatial coding cell in this type of environment. And my question is how the space coding neurons can represent an environment made of this type of connected room. So for this last question, my PhD student, Celia Laurent, is recording the neuron in the retrospinal cortex, which has been shown to be critical to find our way when we navigate between distant rooms. People with disorder of retrospinal cortex cannot move between distant rooms. And Celia is recording this neuron while rodents move in a circular open field. So she takes some picture for you today, and I put the picture in the presentation. So it's in the open field. And she also records the same neuron while the animal move in two rooms connected by a central door, and the two rooms are completely different. And she also records in 4 different connected rooms. And what Celia found is that she recorded some normal edirection cell that I call unidirectional cell in this circular open field. So you have 2 examples of edirection cell. And when the animal is placed and moving in these 2 different connected rooms or 4 different connected rooms, you see that the cell doesn't change their activity. The single directional activity is maintained. She also recorded some non-directional cell in this circular open field. But when the animal is moving in the 2 different connected rooms, this cell become bidirectional. And in fact, this bidirectionality are edirection cells with 2 opposite directions. And these directions are specific to one specific room. So in the direction in red is specific to the room with the red cue card, and the direction in blue is specific to the room in the blue cue card. And she records several bidirectional cells that are active in several opposite directions, as this 3 examples. And when animals were placed in the 4 different connected rooms, the bidirectional cell become chi-directional cell. So we have the same number of directional activity as the number of connected rooms. So the retrospinal cortex contains this multidirectional cell. And our hypothesis about this cell is that they can underlie the global and local representation of connected room. In fact, the cells that maintain the same directional activity between the different connected rooms are cells that code the connected room in a one global environment. So they may code the spatial representation in this global representation of space, while the multidirectional cell can represent this connected room in a local representation of space. So the last type of cell are grid cell. So we are working on in the lab. So the grid cell are found in the medial internal cortex, which is a structure that connects specifically the hippocampus, where we have some place cell I showed you before. So a grid cell is a neuron which is active in different localization in the environment. So the path of the animal is shown in the black line, and I superpose the activity of one single cell. And you can see that this one single cell is active with spot of activity very well regularly organized. And if we look at this organization, it creates a nice grid pattern that covers the whole environment. So it's the activity of one single neuron. And this grid pattern is similar to what we do when we want to navigate in with a map in a space, because we put a grid which serves as a metric coordinate system. So this grid cell could be a metric coordinate system in the brain. So this is an example of a grid cell. So the animal is moving in this square box. And each time you see a white diamond, this is the activity of a single neuron. So the experimenter entered to give some food pellets to the animal to force him to go everywhere in the environment. And after some minutes of recording, you can start to see the spot of activity. And after 20 minutes of recording, you see the grid pattern of this single neuron appears. And there is no specific behavior of the animal. It's freely moving in this environment. So in the lab, we are working on grid cell with Francesca Sargolini to ask the question, how do grid cells represent a place with a different value? So we use a task in which a specific zone in the open field has a high motivational or a high attention value. And we call this zone the goal area or the goal zone. And the animal is doing the continuous navigation task in which he learns to find a specific zone, this goal area. He has to go in this zone to wait 2 seconds in order to have a food that falls randomly in the environment. He is moving to find this food, and after he go back to this goal zone. So this is an example of an animal doing this task. So the goal zone is at the bottom of the video. And you see the animal looking for pellets and coming to the goal zone and waiting 2 seconds. The pellet fall, and he was randomly navigating to find the pellet. And he's coming back to this goal zone, waiting. So in the lab, we record the grid cell when the animal were performing this continuous navigation task. We have also some grid cell when there is no goal zone, so when the animal were freely moving in the environment. So in this condition, we observe, as shown before, a very nice regular grid pattern. And I draw the equilateral triangle here to show you that the distance between the fields are equal in this whole environment. While when an animal is engaged in this continuous navigation task, you can see that the triangle is bigger when the animal is around the goal zone. So the distance between the fields increase close to the goal zone, while the distance between the field doesn't increase far to the goal zone. So in this condition, we have an irregular grid pattern, and we believe that this grid pattern locally expands around the goal zone. This data has been obtained by Fabrizio Capitano, who was a postdoc of Francesca. And we record many of these cells, and we sum the activity of all grid cells to see if the cells were active in all the whole environment. And we observe that the activity of population of grid cells decrease specifically at the goal zone. So we can think that this change of grid cell activity may inform that this goal zone is highly relevant for the animal and for its behavior. So this was my last slide about the experiment, and I just show you some of the publication we did in the team the last 3 years to show that we publish on this 3 type of cell, place cell, education cell, grid cell, but we also do some behavioral experiment about navigation to explain the process of navigation for rodents. So thank you for your attention, and I just want to finish to tell you that with this spatial cognition team, which is amazing, we study navigation. Of course, we investigate this navigation, but we also investigate some other topic related to the spatial navigation, which is memory, planification, and we can also investigate sensory integration, decision-making, robotics, computation, neural network, pathophysiology. So spatial cognition can allow us to do very, very nice neuroscience and to collaborate with very nice other researchers in other fields.
Thanks a lot, Pierre-Yves, for this.
I hope I was not too long. Sorry.
Oh, okay. Sorry.
Finish.
Okay.
Okay, so question.
You have 2 questions in the Q&A section.
Okay, so the first question is, could some rats have more multidirectional cells, and would that mean that they have a bad sense of direction, like myself? More directional cell, in fact, I don't know. It's just preliminary results. But that doesn't mean they have a bad sense of direction, because the animal has, in the same time, unidirectional cell. So both bidirectional and unidirectional cell were recorded in the same, when the animal, in the same time, so similarly. So this, for me, means that the animal can have this 2 types of representation, which is local and global representation. But I have now to do some behavioral experiments to see if this cell activity were linked to the sense of direction and to have a behavioral component.
You have the first question was from Michael, which is at the bottom of the list.
Okay, sorry. What is the reference for head direction? Is it head position versus the wall or versus the mouse body or both? So we don't know what is the reference for head position. When you play the animal, you cannot predict in which direction the cell will fire. But we know that it's an internal representation of direction. So when the animal is moving, the cell is maintaining always the same direction during all its navigation. So I'm not sure I answered the question, but I remember that some computational model say that the one reference could be the gravity with the head position. So the gravity that are perceived by the peripheral vestibular system. And it's not clearly the mouse body. It has been shown that head direction cells are clearly not related to mouse body orientation.
And I'm sorry, we have a question from Lydia Karkarian, which is going to take the, okay, it's okay for you, Lydia? Yes.
Yeah. Okay, thank you very much, Pierre-Yves. It was a really very clear and fantastic job. I have two questions regarding the study with place cells and parental obesity. I was wondering whether the effects you have seen with the decrease in spike density is really a specific effect, or can this be due, for example, to difference in the animal emotional state, anxiety, attention, and so on? So is the activity of these place cells modulated by emotional state?
Okay, it's a very good question. So it could be. It's preliminary results. So with Manon, we are analyze this data, and we know that place cells are linked to emotional or attentional state. So it could be linked to different emotional or attentional state. I know that my animal were placed in the same condition between my two groups. So I believe that my condition doesn't change anything between the animal, but of course, yes, it could be linked to different attentional state. Yes, it is possible. But if it was the case, I would not expect no spiking, but I would expect more activity of the cell that were outside the field, like you can see the red dot in this position, which could be a mistake of the animal position computation. So I would expect that the cell continue to fire, but to have a noisy activity.
Okay. And the second question regarding your interpretation for the absence of change of unidirectional cells, why do you infer from that they might be involved in global representation? I didn't take your point.
Okay, because the cell doesn't change the activity. So when the animal is freely moving between the 4 compartments, the cell between 2 or 4 connected compartments, the cell doesn't change the activity. So for me, it's like the education system include all connected compartments as if they were in one big environment. It's like when you are in the lab, you have the floor of the lab with many offices, and you can move between the office, but you know the position of the office.
The global one, okay. Right. Yeah.
Yeah, okay. Okay, so I'm waiting.
Go back to the Q&A section.
Okay.
Thank you.
Thank you. So do you think that the capacity of sight or other senses have an influence in the distribution of spatial region where there is a lot of activity? Do you think that rodents can orient themselves with the Earth magnetic field? So other senses, I don't know, maybe. The magnetic field, some people are looking for magnetic fields for this type of activity, but we have no evidence about correlation between this magnetic field or other senses than visual information, auditory or tactile information, or also self-motion information. So I'm sorry, but I don't think I can answer. If I have to answer, I don't think magnetic fields is relevant for rodents. So I believe that it doesn't, it isn't useful for this capacity of navigation. It's more something for the birds than for rodents. So we should change the model, the animal models, to look at this type of senses. Okay, did the representation of space is stable in grid cell over weeks? Yes, it's stable. So when you record a grid cell in a specific environment, you can place the animal in the animal facility, and the day after you can record exactly the same cell and you will exactly find the same activity at the same position. So grid cell, place cell, and direction cell are stable within a specific environment. So they are a form of spatial memory also. That's why some people are investigating memory with this type of neuron. Does fat acids inhibit in some way the neuron of directional cell? Okay. I don't know. I'm not sure what is a fat acid. So if people can explain me what is exactly a fat acid, but I don't think it's something I can answer because I have not the knowledge on that. Okay, I switch to another question. Have you investigated grid cell pattern and stability over days in the 4-room environment? So the grid cells are only in the medial entorhinal cortex, and Celia recorded only in the retrospinal cortex, but it's part of her project to look at what happened in the grid cell in this 4-compartment environment. 4-room environment. So you have to wait, I think, 1 or 2 years, and I can answer this question. Sorry. Is spatial representation affected with aging? If yes, is this correlated with cells activity or numbers? So yes, spatial representation is affected with aging. So what is affecting? So Vincent Hogg did a postdoc and shows that aging animals have less reliability of the spatial activity. This means that sometimes when the animal go inside the specific place when the cell could be active, the cell doesn't fire. So there is this boots of path where the cell is not active, and this may underlie some spatial deficit with aging. So we have this type of spatial representation that are affected and could be linked also to the activity of the place cell, for example. Is this correlated with cell activity? Yes, and numbers, I don't know if it's correlated with numbers. I don't think it has been made.
We have a question from Lucas. So
Yeah.
Lucas, if you can take the end.
Yes, it's a little linked with what you already said about people who are looking at that, the spatial cells with memory, but it's about multidirectional and grid cells. Do you have a specific time, a training time before looking at that cells, or you have a different time because you're training the mice before looking at the cells?
So no, we don't train the mice, the rats before looking at this cell. These cells are active the first time the mice or the rat is moving in this environment. And some researchers in London look at this cell in development, and they find that pups have a very nice grid cell, place cell, and education cell. And we don't need to learn about our environment. We have just to move, and this cell start to fire exactly as I shown. And after they stabilize their activity when you have some, when you are far more familiar with the environment.
Okay, thanks.
You said that obese rats had cells that doesn't work as the cell of the normal rats. Yes, it's Yes, some properties are different. So this is what I try to explain, is that if we want to compare the spatial activity, you can see a very nice spatial field. So if we are looking at this color-coded spatial map, the place cell of the obese from offspring parents is as nice as the place cell. So the information of the place cell is similar. But if we are looking at the how many spike, how the spike can compute this information, we have a less number of spike. So less number of spike mean we can have a less spatial information in this condition.
Thank you, Pierre-Yves. And I'm going to pass the end to Gabrielle Gallon, which has a question, and it will be the last one because it's already 5.
Okay, thank you. So Pierre-Yves, if I understood well, you said that you don't know if the other senses are involved, and you don't think that the body position has something to do. So I wonder how in your first arena, the first movie you show, how can they, the first one, the very first one where everything was black and it's a round, it's a circle, so there is no indication, so how can they differentiate a position? How can this neuron be, activated only on this spot because there is no difference anywhere?
Okay, I understand the question, and maybe I didn't answer correctly the other question. The animal is using sensory information, of course. Is using the visual information because there were this object. Is using also the tactile information because if you can see my cursor, I show the object, and the object are different shape and different texture. So one object is white with a different texture to this other object. So the animal will integrate this sensory information from tactile information, visual information, and it will use also the self-motion information to know that he has some angular acceleration or linear acceleration that were given from vestibular system, or also information about his movement from proprioception. So he will integrate all this type of information to know that is in this space. You have also the word that tells the animals that here in this location. You can also compute the distance from the different object to know where he is. So you will integrate all this information inside the hippocampus to create this spatial representation and to have this cell that fires specifically in this position. Did I answer your question?
Yeah, perfectly. But if there were no indication, then the red spot we can see here wouldn't exist.
Okay, so if there is no indication, so the animal can rely to self-motion information.
Okay.
Vestibular system. But this has a limit because self-motion information is not perfect. So at the beginning, you will have a nice spatial activity, but as you suggest, the cell will continue to be active, but the animal will do some error of position.
Okay, normal. Thank you very much.
Okay, thank you.
Thanks, Pierre-Yves. We have just a last one from Miriam Kerr on the Q&A section.
What is the interpretation for enlarged area between two firing spots in grid cell at proximity at the goal place? So this is a good question. And to be honest, at the beginning, we didn't believe that the grid cell will expand because people hypothesized that grid cell were a coordinate metric system. And if we need to compute a goal zone, specific goal zone, which is highly relevant for the animal, we need to better compute the position of this zone. So we believe that this better better resolution of the spatial resolution of the goal zone will be linked to a decrease of the grid cell activity. So we find this increase of the distance between the field. So our idea is that it's not just a single cell that will inform about this relevant goal area, but it's more the population of cell, because when you look at the activity of the population of the cell, you have this decrease of activity. And it's more the decrease of activity at this goal zone that will inform that this area is very relevant. And maybe the medial antennal cortex gives this information to the hippocampus to know and to behave in this environment.
Okay, so thank you, Pierre-Yves, and thanks everyone. So I'm just gonna share my screen. Okay, share. Okay, so our next Neurobinar will be on March 18th at 4 p.m. with Pascal Kilikini from INS and Estelle Nacul, which is a postdoc from Neuroschool, which is working on at Boarding Ring and LNC. So it's a different format. This one, we will not host PhD students, so only a postdoc and a researcher, and the postdoc presentation will be oriented a different way to help maybe PhD students to find their orientation to a postdoc, sorry. And I also invite you to take up the little survey you can find on Zoom and stay connected with Justine Faccini for the AGC3C presentation. And thanks to all for your very interesting interaction.